Okay, good morning, good afternoon, everyone, and welcome to BioMass Canada Cluster webinar series. Today, we are going to have the second webinar from the series. And before that, I just want to mention that BioMass Canada Cluster, this is a cluster that in the first webinar, Don Smith explain the scope and the objective of this cluster. Just a brief background on BioMass Canada cluster. So this is a cluster led by Biofunet Canada Network, and this is financially supported by Agriculture and Agri-Food Canada Agri-Science Program for 5 years. And we have 10 activities. So each of these activities are focusing on one aspects of agriculture biomass to make it a feedstock for bioenergy, biofuel, and other bio-based products. So we look at producing and also processing and distributing the agriculture biomass, as I mentioned again, as a feedstock for various applications. Just quickly introduce myself. My name is Mahmoud Abadian. I'm a research associate at University of British Columbia, working with the Biomass and Bioenergy Research Group, as well as forest product, biotechnology, and bioenergy. And today, as I mentioned before, we are going to have a presentation from another professor at the University of Saskatchewan that I'm going to introduce in a few minutes. But before that, I just want to mention that you can have access to all of the webinars on BioMass Canada cluster website. You just need to go to the website, click on the webinar, then you see the list of 9 webinars. We had one 2 weeks ago on December 4th. You can find the recording and also a slide for that webinar. And we are going to do the same for today's presentation and also the future ones. Just a few housekeeping items. As I mentioned before, we are going to record this webinar, and also we are going to share the slides, so you can find both recording and also slide on the website after the webinar. Also, you can see 2 boxes. One of them is Q&A and another one is chat. Please type your question in Q&A, and I try to ask as many questions as possible from the presenter. And if we cannot answer any of the question because of the limited time we have, I'm going to send a question to the presenter so we can post all of the unanswered question on the website. And also, I encourage everyone to type your name and affiliation in the chat box, so at least we have a sense of social networking in this platform. Okay, so with that, it's my great pleasure to introduce my colleague, Dr. Ajay Dalai from University of Saskatchewan, a distinguished professor and Canada researcher at the Chemical and Biological Engineering Department. So Dr. Dalai has served as head of Chemical Engineering Department and also the Associate Dean Research and Partnership in Engineering at the University of Saskatchewan. In 2001, he was awarded a Tier 2 Canada Research Chair in Bioenergy and Tier 1 in 2009. In 2012, he accepted the position of Special Advisor to VP Research Signature Areas of Energy and Mineral Resource. His research focuses on bioenergy and value-added product from biomass, pollution control, and novel catalyst development for gas to liquid technologies, biodiesel production, hydrogen, syngas production, and also hydroprocessing of gas oil. He has published over 550 research papers, mostly in the heterogeneous catalysis and catalytic processes in international journals and conference proceedings. His groundbreaking research in environmentally friendly processing and the conversion of biomass to bioenergy have led to the over 28,000 citation of his work and an H index of 78. So with that, I give the floor to you, Ajay, and we're looking forward to hearing your presentation.
Okay, just wanted to You can see my screen,
Yes, yeah.
Mehmood. Okay.
Yes, go ahead. Yeah.
Uh, Thank you very much, Mehmood, for kind introduction, and also wanted to thank Shamin from the Biofuel Net Canada for initiating this lecture series as well, and also thank Canadian Agriculture Partnership Program for funding our research and research across this country, especially related to agricultural engineering and science. Uh, so, um, so, I, so the title of my presentation is Novel Biochar and Activated Carbon Productions from Biomass and their Industrial Application. And the funding that we have is for this research, which is called or titled Biochar Production from Canadian Lignocellulosic Biomass and Application for Carbon Sequestration, Soil Improvement, and Crop Productivity. And the focus here is for to pyrolyze and gasify local Canadian agricultural biomass, such as canola meal, canola hull, and oat hull to produce the main fuel products such as biofuel and syngas. And also we are looking at using biochar for soil fertility, crop productivity, environmental absorption, and CO2 capture. And I hope that this research would certainly benefit the environmental as well as the agricultural industry. So I'll not limit the talk to what we are working on now, but just give a little bit overview what we have been working for a while related to biochar and activated carbon areas. So if you look at this slide, the world total primary energy supply, we see that about 81% of energy is coming from fossil fuel, and only 13% coming from renewables, and about 5% coming from nuclear. So because of this large use of fossil fuel around the world, and we all know that when you burn fossil fuel, you get also a lot of carbon dioxide and other greenhouse gases. Over last 40-50 years, due to industrialization and also due to population growth, there has been more energy use around the globe. Therefore, there is a more increase in CO2 emission, which has a direct link with the temperature rise around the planet. And if you look at this particular graph, you see there is a fluctuation of temperature every year. That's because of the fluctuation of CO2 in the environment. What happens in the summer month in the northern hemisphere, where there are more forests, there's more photosynthesis going on by reaction of CO2 with moisture, with the chlorophylls that we have in the plant, and that sequesters a lot of CO2, and then the CO2 concentration in the planet goes down in the summer month in northern hemisphere. And when we have a winter in northern hemisphere, that photosynthesis work is slow, and therefore there is more buildup of CO2, and that is again due to more energy usage in the northern hemisphere during colder months as well. And also, I'm very happy to report that Canada has been a part of Kyoto Protocol, which was signed in 1997. Also has been part of a Paris Agreement, which was signed in 2015. Most of the countries could not really achieve or meet Kyoto Protocol, but according to Paris Agreement, Canada has pledged to reduce its greenhouse gas emission by 30% below 2005 levels by 2030. Developed countries, including Canada, are committed to spending at least $100 billion a year between 2000 and 2025 to help emerging economies deal with the effects of climate change. And that's where the biomass is coming to play. So use of renewable sources such as the biomass is one of the main strategy used in last 2 decades to find solution for some current environmental concern, such as greenhouse gas emission, mercury emission, global warming, soil fertility, etc. There are abundant amounts of waste biomass in North America, unlike fossil fuel, which is not well distributed around the planet. The agricultural biomass and other biomass are more or less uniformly distributed over every country on the earth. And that those are again available with the technologies for conversion to value addition, and also the climate change strategy, meeting climate change strategies as well. So if you look at the biochar and activated carbon area, the sources of biomass and activated carbon are the lignocellulosic biomass or qualified materials, and the production method are physical and chemical activation methods. And in the physical form, they could be granular or powder. They have many applications. I will talk in a minute about that. And if you more interestingly, if you look at the market and price, the production in the world is 300 to 400 million kg per year, and consumption growth rate is about 7% per annum. And 55% of the carbon is available in powder form and 45% in granular form. So the granular activated carbon sells about 1.65 to about $10 per kg, whereas the powder activated carbon sells for 80 cents to $2 per kg. So if you look at the biochar properties and application and follow this red arrow, so we can produce biochar varying from temperature from 300 to 700 degrees Celsius, and with that we can increase the aromaticity, increase the ash content, the pH, the surface area, and pore volume. And in the other way, when you go from higher temperature to lower temperature, we can increase the yield, volatile content, conductivity, and cation exchange capacity. So the one that you see in the right side, the biochar has many applications. Start with the fuel application. It has a lot of heating value. It has an agronomical application, such as water retention, plant nutrients, and soil conditioner. You can also apply for carbon credit. We can also apply for making specialty materials such as biocomposite, etc. And we can also use it for biomedical and pharmaceutical and gas storage applications. And also we can use it for catalyst, adsorbent, and water treatment, etc. So it has a very wide variety of applications, and that are all coming from biomass, either agricultural, municipal, or forest biomass. So if you look at this slide, the property of biochar itself not only depends on the pyrolysis condition of the biochar, but also depends on the nature of the precursor itself. And also the properties of the activated carbon not only depends on the origin of the biochar, the quality of biochar, but it also depends on the activating agent and conditions to provide a porous physical and chemical structure for this material. So here is a photograph of a fast pyrolyzer, where we have a hopper which takes the biomass, powdered biomass, into the fast pyrolyzer, and it pyrolyzes. The pyrolysis time is roughly 1 to 2 seconds, and then the vapor with the particles are taken to a cyclone where the particles are separated, and that's where the biochar gets deposited. And then the vapor is taken to a condenser where the liquid is condensed, and also the gas can be cleaned and recycled for this pyrolyzer. And the yield of the biochar from the first pyrolysis process is roughly 10 to 15%. So with this introduction, so let me talk about the environmental applications of bio-based activated carbons produced by physical and chemical activation methods. So if you look at this particular slide, we see that biomass through pyrolysis, we can get a biochar, and through physical activation, for example, with the carbon dioxide and steam activation process, we can get activated carbon, and also through chemical activation with phosphoric acid, zinc chloride, and potassium hydroxide, we can also get an activated carbon. So if you look at this particular slide, so the physical activation, when we conduct, we have to manipulate the 3 parameters such as activation temperature, mass ratio of steam to char, and activation time. And for CO2 activation, we have to manipulate the activation temperature to and activation time. So that these have a lot of impact, or large impact on the yield and product quality. In case of chemical activation, for example, with potassium hydroxide, we mix potassium hydroxide with the char before activation, especially with inert gases such as nitrogen at a certain temperature. So this gives a typical elemental analysis of biochar from fast pyrolysis process, where carbon is roughly 80 to 83%, and it has about 10% or so of oxygen, and it has a ash content of 3.4%. So if you get activated carbon from biomass, of course, the ash content will be low. But if you get from coal, for example, the ash content is very high, and the quality would be really, really low. So this one gives a schematic for activation setup, where you have a tubular reactor where we load the biochar, for example, and heat it with nitrogen. And at the activation temperature, we start feeding the steam at certain flow rate, and let the activation continue for a certain time. And then we collect the tar and also the gas. And then after that, we cool it down to room temperature and look at the quality of the activated carbon. So what are the process parameters that we usually look at for activation through physical activation of biochar? Activation temperature, steam to char ratio, and activation time. You can see the range that we usually operate, and to get a very high quality product. The heating rate for the tubular reactor is roughly 3 degrees C per minute, and we have certain nitrogen flow to take the volatiles, etc., out of the reactor. And each batch we use roughly 20 grams of biochar. And for chemical activation again, so we have the activation temperature, KOH to char mass ratio that we vary, and activation time also we vary. And then again, these are all batch processes. So this is a very interesting slide in the sense we want to know what reactions are involved in steam activation of biochar. Here is a carbon reacting with water to produce hydrogen and carbon monoxide, and it's an endothermic process. And this product, carbon monoxide, again reacts with the feed steam to produce additional hydrogen and CO2. And also the product hydrogen and CO can react together through a methanation process to produce methane, for example. And these two reactions are exothermic in nature. In case of potassium hydroxide activation of biochar, potassium hydroxide reacts with carbon to make the material porous, thus producing potassium, hydrogen, and potassium carbonate. So typically, when you have a 3-parameter to optimize, we use a central composite design method, which leads to 20 experiments with 5 central points, which are used to get the average and statistical calculation, including the variance and standard deviation for the data. And through process optimization or the central composite design method, we can optimize the temperature, steam to biochar ratio, and reaction time for steam activation, and reaction time and temperature for CO2 activation, and reaction temperature and potassium hydroxide to biochar ratio, and flow rate of nitrogen for the KOH activation process. Here the target is to produce biochar at least or the carbon which with a yield of at least 50% and surface area of at least 500 square meter per gram. And just to remind you that the biochar that we get from the first pyrolyzer, the surface area is usually less than 10 square meter per gram. So based on this process optimization study, you can see that you can relate the surface area and yield with the temperature. Here the steam to carbon ratio and reaction time. And you can see there is also interaction between temperature and the steam to carbon ratio when we calculate the surface area as well. So the beauty of this equations are that without performing experiment, if you want to set certain temperature and other parameters within the ranges that we studied, we can calculate the yield and surface area for the material. And you can see this 3D plot, how the temperature and mass ratio influence the BET surface area at activation time of 2.5 hours for steam activation process. Here you can see at optimum temperature and also steam to mass ratio, we can get a BET surface area of about 900 square meter per gram. Whereas in chemical activation, when you vary the mass ratio of KOH to biochar and temperature, you can see that we can get a surface area of over 1600 square meter per gram. And here you see that the temperature has somewhat positive impact, whereas the mass ratio, it has a firstly positive impact, then the impact is a little bit negative. So that's where the optimization is required. So here, once you optimize the conditions to produce high quality biochar with the high yield, then we have a pilot scale setup where we have got a reactor and a superheater, have a boiler. And so we use this system to generate large amount of activated carbon when needed. And here is the picture of that pilot plant where we have got this boiler, and then we boil the steam at about 82 psi and 300 degrees C, and pump it to superheater to heat it so 700 degrees C. And the superheated steam goes to the reactor containing the biochar, which is roughly 5 kilograms in comparison to 20 grams in smaller reactor. And then we can get about 2.5 kilograms of activated carbon through this process per day. So now with that information, let me switch to some of the applications. So we have been working on the biochar application or bio-based activated carbon for many different processes. So we have 5 processes here we have given. One is the removal of hydrogen sulfide from gas stream, removal of mercury from the flue gas in coal-fired power plant, removal of carbon dioxide from flue gas stream, and application for improving the soil quality for plant growth. That's the funding that we have now from the Biofuel Net and the Canadian Agriculture Partnership Program. And also we have done work in the removal of ammonium ions from liquid streams. So just to the first one, the hydrogen sulfide capture. Just wanted to give a little bit of background that when you look at any sewage treatment plant, the anaerobic gas or digester produces a gas with about 50% CO2, 50% methane, and it also contains, the gas contains 1,000 ppm to 10,000 ppm of hydrogen sulfide. Typically, this gas is burned to produce power or the heat for the plant, but during heating, the hydrogen sulfide produces not only sulfur dioxide, but the saturated gas also produces a lot of corrosion within the plant. So the idea there is to remove the hydrogen sulfide completely from the gas stream so that the gas is more clean. It won't produce the acid, the sulfur dioxide for acid rain, or it will not. It will also prevent the corrosion happening within the plant. And the hydrogen sulfide is also present in the gas wells when you produce oil and gas. And if the hydrogen sulfide concentration is less than, say, 1%, the closed process becomes ineffective. So this process is quite profitable. So here we have 4 different materials: the physically activated carbon prepared from biochar, and then we acid treated that, and we are thermally dissolved at 750 degrees Celsius. And also we produce the chemically activated carbon using KOH from biochar. And in this table, so with these 4 different materials, we can see the surface area varies from 640 to 836 square meter per gram. The total pore volume also increase when you do the thermal desorption and chemical activation. So here is the hydrogen sulfide oxidation experimental setup. What we do is we load the activated carbon in this reactor, and we have the sour gas coming from 1% H2S in nitrogen, and we add stoichiometric amount of oxygen from air to oxidize the hydrogen sulfide to sulfur and water. And most of the sulfur stays within the bed. There may be some carryover of sulfur to sulfur condenser, and then the water that is a byproduct or the main product goes through a dryer, and then do the GC analysis to figure out how much hydrogen sulfide we have removed in the process. So here are some results. You can see the breakthrough time, which is the time at which hydrogen sulfide appears in the product stream. So you can see that in this, when you load, so the concentration is 10,000 ppm of hydrogen sulfide, and oxygen to hydrogen sulfide ratio is 1.05 times the stoichiometric ratio. And we have the hydrogen sulfide stream is half a liter per minute. And we find that the breakthrough time in some cases varies from 90 to 141 minutes. And during that period, you can see in the third case, where you can load up to 90 kilograms of sulfur on 100 kilogram of carbon over a period of time before it deactivates or the hydrogen sulfide appears in the gas stream. So this is a really very useful process to apply the activated carbon to take some hydrogen sulfide from the gas stream. So now let me switch to the removal of mercury from the flue gas in coal-fired power plant. If you look at this pie chart, you find that the electricity generation produces about 34% of mercury, which comes from the coal, and Canada emits roughly about 7,000 kilograms of mercury per year. And this mercury from different units, they'll fall in the soil and water, and through the soil it can also get through food to us for consume that mercury, or through water it can go through the fish and come back to us to consume as well. So if you can remove this mercury from the source, it's really very useful thing. And when you look at the mercury emission from the power plants, the mercury content in coal varies from 0.01 to 3.3 parts per million. And here is the combustion that happens, which produces the elemental mercury, and the mercury in parts per billion, 1 to 35 parts per billion remains comes under in the flue gas in form of elemental mercury, oxidized mercury, and particulate bound mercury. So what we did is in the laboratory, we basically took mercury and vaporized it and passed through a bed of carbon. And we have a mercury analyzer to see how much mercury is being removed through this process. So what we did is we varied the temperature from 25 to 165 degrees C. Because the adsorption is an exothermic process, lower temperature works better. You can see that we can remove more mercury at room temperature. Then what we did, we functionalized the activated carbon with halides. For example, with chlorine, bromine, and iodine. And we find that when you add about 1% iodine, it can remove 100% of mercury from the gas. And then what we did is we looked at potassium iodide and vary the concentration from 0.1 to 1% in the activated carbon. And we find that with 0.5 to 1% of potassium iodide, we are able to remove 100% of mercury from the gas stream. And here also 0.1% ammonium iodide does work also very well. And then we use that functionalized material at a different temperature. And then you find that for the functionalized material with halides, or especially potassium iodide, we require about 135 to 195 degrees C to take out 100% of mercury from the gas stream. And then we applied this technology to the power plant, which is the emission control research facility at Coronac, at the SAS Power plant in the south of Saskatchewan. And we had a slipstream that we injected our activated carbon, functionalized activated carbon. And then downstream, it had also a mercury analyzer. So this we figured out that we have to feed in this activated carbon when the temperature is roughly 130 degrees Celsius. And you can see that we are able to remove mercury with the functionalized activated carbon from biochar as compared to the reference material. And this project was implemented by SAS Power a few years ago, and they spent about $34 million to scale up and apply this technology to remove the mercury from the flue gas that comes out from the stack at their power plant. And we also did some synchrotron work at the Brookhaven National Lab to find what exactly is happening. And we find that the mercury cations, basically, they are attracted to these halides or sulfur, which are attached to the activated carbon biochar surface. What that means is if you don't have halides or sulfur, this technology will probably not work. And fortunately, the coal has a lot of sulfur, and those particles or the flue gas is a lot of sulfur, and the sulfur gets attached to the activated biochar surface, and that's what attracts the mercury to catch onto. And all this process happens in less than 2 to 3 seconds after the exposure of the active functionalized activated carbon to the flue gas stream. And now let me switch to the carbon dioxide capture research that we have been working on. So here is the fuel and air. When you burn fuel, we not only produce a lot of energy, but also produce the carbon dioxide. We also produce NOx and SOx and hydrocarbons through this process. And in order to capture that carbon, we can basically look at the adsorption on activated carbon. This technology has potential because of low energy requirement, and it's a cost effective, and also it can be applicable over a relatively wide range of temperature and pressure. So what we did in this process is we optimized the biochar for CO2 capture. So you can see the all these activated or the optimized conditions for steam, CO2, and potassium hydroxide activation. And then we looked at the activation yield. You can see it varies from 35 to 59%, lower yield with the chemical activation process and higher yield with the physical activation process. But nonetheless, with the chemical activation process, we can get a material of 1400 square meter per gram as compared to 820 to 840 for the physical activation process. And the pore volume is also 0.62, which is higher. And also you can see that ash content is also lower in this particular case. And in all cases, the carbon content varies from 85 to 92%. And then what we did is we looked at, measured the pore size of this material. So you look at the ultra micropore volume less than 0.8 nanometer, and super micropore volume between 0.8 to 2 nanometer. And here we have the total volume as well. So you can see that for the chemically activated carbon, we have about 0.39 cc per 0.39 cc per gram, whereas the total volume is 0.62 cc per gram. So we have a lot of micropore activated carbon available with this chemically activated, chemically obtained activated carbon from biochar. And we find that this micropore is very, very useful for this particular process. And with the central composite design, we also performed experiment at different temperature and CO2 concentration in the feed stream. Here are some of the data. So look at the impact of, so here is the adsorption capacity in milligrams of CO2 per gram of adsorbent for KOH, CO2, and steam activated carbon at a different temperature. So if you look at any particular temperature, we find that the chemically activated carbon is more suitable for this process, and lower temperature works better because it's an exothermic process. So lower adsorption temperature works better. And when you go to much lower temperature, about 16.7 degrees C, we really did not see any benefit. And also when you cool it down, you also use energy to do that. So this concludes that at room temperature, we are able to remove 75 milligrams of CO2 per gram of adsorbent at room temperature with activated carbon produced from biochar using potassium hydroxide as a chemical reagent. And so the way it works is you have a pressure swing adsorption. When one adsorbs, the other one desorbs. So that's how you go from one to the other, and that gives a very high pure carbon dioxide for other applications. So here are some of the process optimization for CO2 adsorption. We have CO2 concentration and temperature varying for steam activated carbon and KOH activated carbon. And if I see that at optimized condition, here the removal is 60 milligrams per gram. Here is about 20% higher. So that is 80 milligrams per gram for KOH activated carbon. And let me conclude this part of the presentation. So what we find is biochar is used as a precursor for production of physically and chemically activated carbon. And there is a very strong correlation between surface area and yield with the operating conditions. Also, we have used a pilot scale unit for steam activation process for producing large quantity of carbon for industrial application. Also, we have used biochar based activated carbon for removal of pollutants such as hydrogen sulfide, mercury, and carbon dioxide from gas stream. So the second part of the presentation deals with the production of biochar using slow and fast pyrolysis techniques in stationary and mobile pyrolysis units for agricultural applications. So this is a very important slide. I'll spend about a minute on this. So role of biochar in soil improvement. You can see that biochar contains alkali and alkaline earth metals that can neutralize acidic soils. So it's very, very important. Also, it contains NPK that can help supply elemental nutrients to plant roots. Also, biochar forms a complex with soil and plant roots to prevent soil erosion. And also, biochar has a satisfactory water retention and also the fertilizer retention ability to keep the soil healthy and moist. Also, biochar enhances many soil properties such as texture, fertility, water content, salinity, nutrient content, and organic matter. Biochar also holds the stable carbon in the soil, helping the carbon capture and sequestration. And biochar promotes crop productivity, and biochar can hold beneficial bacteria and fungi within its porous structure to continually supply plant growth hormones and aid in carbon and nitrogen fixation. So we have started this research in collaboration with the Agriculture and Bioresearch College in the University of Saskatchewan. So in this, I'm going to show what we have done so far. So we have we have used slow pyrolysis of canola meal and looked at the effects of pyrolysis temperature on the product properties. And here is the pathway. So we have a precursor. So slow pyrolysis and fast pyrolysis look at the quality of the biochar and use the KOH activation to produce activated carbon. So in the slow pyrolysis, we have used, as I mentioned, the canola meal in a fixed bed system. The pyrolysis temperature varied from 300 to 700 degrees C, and we have a nitrogen flowing through the system at a heating rate of 7 degrees C per minute. And canola meal is provided by Fungi Oil Industry in Saskatoon. And we also obtained the weight percentage of oil, carbohydrate, protein, fiber, moisture, ash content in the original canola oil. And we also use the succinate extraction to reduce the oil content from 3.2 to 0.3%. And we also characterize the biochar using different physical and chemical techniques. And bio oil and product gas were also analyzed and to see their potential for value-added applications. So if you look at the thermogravimetric study for biomass, it gives the thermal stability of material. We find that up to 200 degrees quite stable, and then it start degrading after that. So if you look at the approximate analysis of biochar produced from de-oiled canola meal, of course, this has still roughly 0.3% of oil, and we produce biochar varying from 300 to 700 degrees C. And we find that when you increase the pyrolysis temperature, there is a reduction in moisture, and the increase in ash content, and also fixed carbon. Also, there is a reduction in volatile content of the in the biochar. And also, when you start with the same particle size, as you increase the pyrolysis temperature from 300 to 700 degrees C, the particle size reduces with time and with temperature. And also we find that with increase in the biochar or the pyrolysis temperature, we are able to maintain more alkaline metal and other essential element. And of course, there are some heavy metals present in the biochar. So this is a very important slide in the sense that we have plotted the atomic hydrogen to carbon ratio to and atomic oxygen to carbon ratio. We find that with increase in the pyrolysis temperature, we are able to increase the carbon content and reduce the oxygen and hydrogen content. Therefore, you can see the trend here as compared to the canola meal, where the spot is here. Here is hydrogen to carbon is 1.6, and oxygen to carbon is about 0.6. So we did the X-ray diffraction analysis of canola meal and biochar prepared at different pyrolysis temperature, and we find that the broad peak reflecting the cellulose. And also, when we increase the pyrolysis temperature, our ash content is increased, so we get some alkaline and other materials present in the sample. So here is the elemental analysis of biochar, as I mentioned. With increase in the pyrolysis temperature, we are able to increase the carbon content from 48 to 82%, and there is a reduction of hydrogen and oxygen, and also it's no impact on the nitrogen and sulfur content in the biochar. We also looked at the heating value for energy application purposes. We find that we are able to increase the heating value to between 26 to 27 megajoules per kg through this process, pyrolysis process. And the ash content was increased as one would expect. The electrical conductivity decreased, and the pH of the biochar increased, and that's really good for acidic soil applications. So we also did the fast pyrolysis of Canadian waste biomass using a mobile pyrolysis unit that is procured by Saskatchewan Research Council from Advanced Biorefinery Inc. And we varied the temperature from 4 to 550 degrees Celsius and used wheat straw, flax straw, sawdust, and poultry litter. And we also characterized the feedstock and the biochar and also the oil and the gas, all that. So if you look at this particular slide for the biomass characterization, it shows the cellulose, hemicellulose, and lignin content. As I mentioned, when you start heating it beyond 200 degrees C, that's a degradation of cellulose and hemicellulose through the pyrolysis process. So here are some of the data we have. So in increase the pyrolysis temperature for all these four materials, we find that the heating value is increased, so as the pH, and so as the ash content in all the cases. And the surface area of all biochar was less than 500 square meter per gram, and it's quite highly non-porous, as you see this. We also did the ultimate analysis of biochar, where we measured the nitrogen, carbon, sulfur, hydrogen, oxygen content, and this oxygen content was obtained by difference. So you can see by increase in temperature, we are able to increase the carbon content, and here in this case, reduce the sulfur content, and reduce the hydrogen content, and also reduce the oxygen content. So with this, we have also done more work in this area, and because of lack of time, I'm not able to present. But up to this point, I just wanted to conclude that slow pyrolysis of canola meal was investigated at different temperatures, and products were comprehensively characterized. Fast pyrolysis of 4 types of Canadian bioresidue was investigated at 3 different temperatures in a mobile pyrolysis unit, and all the products were characterized for their physical and chemical properties. And we found that the biochar characterization led to our understanding that it has a good application for environmental issues, and also they have a high potential for agricultural applications. So this slide shows what we have done so far related work with the biochar. So we have done some work on activated biochar for mercury capture funded by SRC, NSARC, and WED. So that is completed. Activated biochar for hydrogen sulfide funded by NSARC. I presented just few slides on this. And activated biochar for organic chemical capture from industrial wastewater. So we had a project from industry with the Minetax and NSARC, and that is completed. And activated biochar for biofiber production, that's also completed, funded by NC, led by University of Toronto. And activated biochar for soil application, so that's one we're working, funding from BiofuelNet, and also the Canadian Agriculture Partnership Program and Agricultural Development Fund, which is part of the Ministry of Agriculture in Saskatchewan, and also industry. And activated biochar for carbon dioxide capture from industrial flue gas. This is also ongoing as this last but one project funded by NSARC, Canada Research Chair Program, and university. So over years, I have collaborated with Professor Don Smith at McGill University, Professor Franco Beruti at Western University on these projects, Professor Andrei Majak, UK Biochar Research Center, University of Edinburgh, and Professor Raphael Okon, Harriet Watt University, Edinburgh, and Dr. Stanley Suchak and Ranganathan from Saskatchewan Research Council. So though they are were very helpful in early days when we started working with SaskPower on mercury capture, SaskPower and Titan Energy, New Life Green Tech, and Hey Holding in Montreal. So this is I think very important slide where most of the funds came from. Government of Canada under the Canadian Agricultural Partnership, BiofuelNet Canada, Saskatchewan Ministry of Agriculture, NSARC, Canada Research Chair Program, Western Economic Diversification Canada, and we also did a lot of collaboration with Canadian Light Source and Saskatchewan Structural Sciences Center, and SRC, SaskCanola, and our current industrial partners are New Life Green Tech and Hey Holding. So with that, I just wanted to thank you all for your attention, and thank you for your time. And I hope I shared some of the work that you may be interested to pursue further. We have lots of publications we can certainly share with you if you are interested to understand more. And because of lack of time, just wanted to wind down at this point. And we have still several minutes. We can go through a question and answer. And before I close, just wanted to share this slide where you see a lot of snow on the top slide. That's what we had in Saskatoon in the winter months, and then the bottom slide, part of the slide shows the scenic Saskatoon in summer months. Thank you so much for your attention, and I'll be happy to discuss further on this. Again, those who attended, thank you from bottom of my heart, and have a Merry Christmas, Happy New Year, and Happy Holidays. Thank you again.
Thank you very much, Jay. I really appreciate rich. It was a information-rich presentation with lots of good application for biochar and activated carbon. We have a few minutes. I know that you have a meeting and you have to go to your meeting, but I guess we have about like 4 or 5 questions. So let's see if we can handle all of these questions. Otherwise, we're gonna send this question to you so you can actually answer this question offline. So the first question is from Maria. She's asking about, I guess this is the first part of your presentation. She's asking about, are the activation results shown for biochar produced from which feedstock? Is it a canola meal or hulls or any other type of feedstock?
Yeah, we actually, Maria, thank you very much I think I know Maria for a long time, and I'm so glad that you attended the presentation. I saw your little note in the beginning. Thank you. When we started our work, we all started with the forest residue and gradually moved to the agricultural residue. And our current focus is more on agricultural residue. But initially, when you asked with the SAS power, those days, those are most forest products that we applied for the mercury capture work.
Okay,
And
thanks.
for the hydrogen sulfide and for the CO2, they are mostly the biochar from the agricultural materials
Okay,
and
thanks.
locally grown agricultural bio residue.
Okay, thanks. And the second question is from Gary Walker. He's saying that I'm a distributor for biochar now in Ontario and would like to know if you see a role for biochar in organic compost, such as poultry litter or like other waste product.
I think that's a really good question. I think Titan Energy in Saskatchewan is producing biochar for a lot of applications. So Gary might know Jamie who owns Titan Energy. So we have not really done a lot of work in for the application that Gary is referring to, but he can talk to Jamie in Titan Energy and see if he has some products in bulk to supply to him to Ontario through his company.
Mhm. Great. So Gary, you could be in touch with AJ. You can find information
Yeah.
on the AJ website. So if you want to contact him, you can actually send an email to AJ. Another question from Mario, AJ. She's asking, what are the next steps for the canola meal pyrolysis work?
Yeah, that's really good. What we are doing now is because we have optimized the pyrolysis condition, we know which ones are more useful for soil application. So we have supplied some of the material to our our colleague in College of Agriculture for soil application to grow some plants, and that work just started. And so our focus for next couple of years is to apply our material for agricultural research. So that's where we're heading to. And we have, as I showed, we have a large production unit. We have a smaller units. So depending what is required, I will supply. And I don't have a lot of expertise in the soil work. So and we have soil scientists part of the team to help us out in that work.
Okay, thanks. Let's see. I guess we have time for maybe two more questions. So Dan Charbuno, hopefully I'm pronouncing his family name correctly. He's asking, are there any commercial installation for creating agricultural biochar in Canada yet, either batch or continuous process?
Actually, as far as I know, there is none, and we actually myself, Franco, and also Don. So I think some of us are basically in forefront in the biochar research, and it'll be really nice if anybody takes this to the field and produce in large scale. We have enough know-how to do that, but there has not been any interest to create a large amount of biochar from agricultural residue so far.
Okay, good. Thanks. And I guess the last question is going to be about, again, hopefully I'm pronouncing the name correctly, Gislian Poisson. He's asking, I'm confused in the difference in surface areas expressed as square meter per gram between early slides and later slide. You showed values of around 1,000 and later five of biochar. Is there really such a variability in biochar surface areas, or is there a mistake somewhere?
Actually, that's a really good question. I think as I showed in one of the slide, the quality of the activated carbon or biochar not only depends on the activation condition, also the origin of the biomass itself. So the variation that you see is basically due to variation in the precursor, number one, and the variation in the process that we use to produce this biochars or the activated carbon. So we have produced activated carbon with the surface area from 500 to 3,000 square meter per gram from the biochar with the surface area of 10 less than 10 square meter per gram. And so every precursor requires some sort of optimization. So the numbers that you see would be optimized number for different biomass or biomass precursors. So that's why those numbers are so different.
Okay.
And
Good.
also want to add, Mehmood, that if you want to have a water application, you have a large pore and a small surface area, maybe 200-300 square meter, 400 square meter will do. But if you want to have a gas application, you need very high surface area with very tiny pores like micro pores or ultra micro pores, as I showed for a CO2. CO2 has a molecular diameter of 0.3 nanometer, and any size that is less than 0.8 nanometer works really well for CO2. But for water application, you need a very low surface area with a very large pore diameter, so that the organic chemicals can penetrate and easily stay, and it can clean the wastewater.
Great. Thank you very much, Jay. I really appreciate your detailed presentation. I'm sure it triggered a lot of discussion and question for the attendees. And I want to really appreciate the time that attendees today to attend this meeting, participate, asking question. And I'm hoping you can attend the next webinar that's going to be January 7. And we are going to have another presentation as part of the activities in BioMass Canada cluster. And I wish everyone a Merry Christmas and stay safe, and hopefully see you in January 7 for the next webinar. Take care and have a good day.
Thank you, Mehmood, for coordinating this. Really appreciate it.
My pleasure.
Thank you.
Bye, everyone.